about 10% of the affected people have a delayed set of symptoms. Normally, the symptoms appear before 6 months of age, and if you have a delayed symptoms, it occurs from 1 to 10 or even into adulthood. Diagnosis of it, the doctors identify ADA during the mother's pregnancy. They take a tiny bit of tissue from the amniotic sac and test it, or they can look at enzyme levels in the fetal blood sample from the umbilical cord. Once the child is born, doctors can test a sample of his or her blood to see if it contains ADA. Another evidence that they look at is low levels in lymphocytes, which are the white blood cells. If their tonsils, thymus, or lymph nodes are absent, or they have less than 1% of adenosine deaminase activity, they can also look at x-rays to see if they have bone abnormalities. Signs and symptoms, like I said, they develop before 6 months of age. Normally, affected babies also are diagnosed with SCID, which is severe combined immunodeficiency, which we'll talk about that later on. Babies are normally more vulnerable to bacterial, fungal, and viral infections. A normal baby can get infections, but if you have ADA deficiency, the baby can have the infection more severe. Some of the symptoms include chronic diarrhea, pneumonia, other lung infections, slowed growth, skin rashes, absent tonsils and lymph nodes, bone abnormalities, and developmental delay. Treatment, there is no real treatment for ADA deficiency, but doctors try to restore ADA levels to improve the immune system function so they can fight off the infections. They do this by bone marrow transplantation, transfusion of red blood cells containing high levels of ADA, enzyme replacement therapy, and also gene therapy. Other ways to treat the ADA deficiency is they can use prophylactic medications to treat pneumonia, or they use intravenous immunoglobulin to boost the body's natural response to infections. The most effective treatment is transplantation of blood-forming stem cells from the bone marrow of a healthy person. Prognosis depends on how severe the condition is and when they diagnosed it and whether the treatment is effective. Without the early diagnosis and treatment, a baby can die before the age of 2. If bone marrow transplant or other treatments are effective, then their prognosis is typically positive.
Alright. The gene affected by this disorder is 20q13.2 gene, so it's basically on the 20th chromosome, and it's exactly between base pairs 44,619,512 and 44,652,232. So it's a gene that they have pretty much know the exact location of. But there are more than 70 gene deficiencies in this disease, most of which involve a missense mutation, the blocking of one amino acid for another, and the adenosine deaminase enzyme. Others, that's why the disease can vary in severity. The ADA gene provides the genetic information to produce an enzyme, adenosine deaminase, which is found at high levels in lymphocytes. The enzyme functions to eliminate a molecule called deoxyadenase, which is made when DNA is broken down, and it's toxic to the lymphocytes. And the adenosine deaminase turns this into deoxygenase. And it's basically the dysfunction or the deficiency in normal cells allows for the toxic compound to build up and therefore hurts the lymphocytes.
Okay, the McCoos number for this disease is 102700. I know it starts with a 1, and usually that means that it's inherited, the inheritance is dominant, but it's actually autosomal recessive. The normal function of the gene, as Nicole said, is to produce a functional ADA enzyme, and this enzyme converts toxic deoxyadenosine to non-toxic deoxyadenosine. The The ADA enzyme, they're found in, all of our cells, but they're most commonly found in lymphocytes, and, the lymphocytes are part of the immune system, and they help to protect against, infections from viruses and bacteria. When an individual inherits an infected copy of this gene from both parents, the individual will suffer from ADA deficiency, which can ultimately lead to SCID. Now, I don't know if that's how it's pronounced, it's either SCID or SCID, but that's I think it's SCID. Yeah. Okay. Mode of inheritance is autosomal recessive, and autosomal recessive mutation is a mutation caused by two mutated copies of a particular gene in an autosomal chromosome. ADA deficiency is an autosomal recessive disease where affected individuals are homozygous recessive. Homozygous recessive individuals have two mutated copies of the ADA gene. The parents of the affected individuals are heterozygous, meaning they are carriers for a mutated copy of the gene, but they are not affected by that mutation. At conception, each sibling of an affected individual has a 25% chance of being affected, a 50% chance of being a asymptomatic carrier, and a 25% chance of being unaffected and not a carrier. This disease is very rare. 1 to every 200,000 to 1 million newborns are affected by the disease. It's really rare. And 15% of ADA deficiency cases lead to SCID. I'm sticking with SCID. Okay. Severe combined immunodeficiency, also known as SCID, is the result of ADA deficiency. SCID is most severe case in infants because it leads to early death. Patients with SCID are prone to many opportunistic infections, and it's kind of like similar to like patients who have like HIV, where the immune system is compromised. So like common infections that a normal person would able to like overcome will be like detrimental to their health. An interesting fact here is that SCID is also known as the bubble boy disease due to the extreme vulnerability to infections. This is a picture of a young child here in the bubble. And I think we have a short video clip we want to show you guys.
There's a disease called X-linked immunodeficiency, which is found in 1 in 100,000 to a million live births.
And it basically leaves patients without the ability to fight infection. More commonly, the public would refer to this as bubble boy disease.
These patients had to be kept alive by basically sequestering them in these plastic bubbles because they have no immune system, and therefore the only way to keep them alive was by not allowing them any exposure to any outside pathogens of any kind. What we did is we got cells from a patient that had this disease, and we converted them to what is known as iPS stem cells. These are basically cells produced in the lab that can then produce any other type of cell that the patient might require for therapy. We have corrected the genetic mutation that causes the disease in this patient, and then gone ahead to produce one type of the immune cells that this patient is missing. And the hope is that we can use those cells for blood transfusion for these patients that would help them manage and treat the disease, which is otherwise incurable and terminal in these very young infants.
A stem cell is a very useful research tool because we can propagate these indefinitely in a dish in the laboratory, and they also have the capacity to become any cell in the whole body. So now we have a cell type that we can use to generate, in theory, an unlimited number of cells that could potentially cure a patient. So for this particular study, we focused on taking these stem cells and changing them into immune cells.
So one of the holy grails of the field is to be able to make a stem cell which is capable of making all of our blood cells and all of our immune cells. We have come very close we can make all the cells which are involved to make the blood. But we haven't quite learned as yet to make all the cells involved in the immune system. That will have an enormous impact on enormous number of diseases. Well, now we have come one step closer.